Welcome back to the Deep Dive. This is the show where we really get into some fascinating, impactful science, trying to distill these complex ideas into insights that, well, frankly matter for your life.
That's the goal.
And today, we're embarking on an exploration that I think might just fundamentally change how you think about your own health, maybe your family's well-being, and perhaps even the legacy you leave behind. Just imagine this for a second. What if the choices your grandparents made, maybe their diet, how active they were, even the environment they lived in. What if that literally left a deep, lasting mark on your health right now in your future?
It's a pretty mind-bending idea, isn't it?
It really is. We're talking about something that seems to go way beyond the DNA you inherited. Something, well, dynamic and incredibly responsive to life itself.
That's exactly right. And that's our mission for this deep dive. We're going into the really captivating world of epigenetics and exploring its profound, undeniable connection to aging. We're going to try and unravel how your unique epigenome, we'll define that, of course, is constantly changing throughout your life. And it's influenced by, well, practically everything.
Like what specifically?
From the food on your plate to your exposure to environmental factors, pollution, stress, even the diseases you encounter along the way.
Right.
And crucially, we want to unpack what all this means for how we understand and approach not just healthy aging, but also that whole spectrum of complex chronic diseases that challenge us today.
So this is kind of a shortcut to the real cutting edge of personal health.
You could think of it that way, yeah. Getting you up to speed on where the science is heading.
And I imagine for anyone interested in or maybe even involved with the genomic medicine program, this deep dive is going to hit home.
Absolutely. It really underscores how understanding these remarkably dynamic changes to our biology, these sort of subtle molecular conversations happening inside us constantly, how that's rapidly becoming central to the future of personalized health.
It feels like a real shift away from that old one-size-fits-all model.
Yeah, completely. Towards something far more tailored, more effective, and crucially, putting a much stronger emphasis on prevention.
So it's clear it's not just about the static blueprint of your genes anymore, is it? That fixed code you inherit.
Not at all. Not by a long shot. It's really about how that blueprint is actively read and used in every single one of your cells every single day. That's where the real story, the dynamic story, unfolds.
Okay, so let's start there then. This deep dive needs a solid foundation. Let's talk about this dynamic duo, the genome and the epigenome.
Good place to start.
All right, so first, let's just recap the genome, your unique biological blueprint. As I understand it, every cell in your body, except for sperm and egg cells, contains the exact same complete set of DNA, right?
That's correct. Your entire genome. Think of it like a master architectural plan for building an incredibly complex house. Every single room, the kitchen, the bathroom, the living room, the bedroom, gets an identical copy of that comprehensive plan.
The full blueprint.
The full blueprint.
Okay, but here's the twist, and this is where it gets interesting. Despite every cell having that identical master plan, your body have over 400 different types of tissues and cells.
That's right. Think about a kidney cell versus a liver cell or a skin cell or a neuron in your brain. They all started from that very same genetic blueprint.
But they look completely different, and they do totally different jobs.
Exactly. And that's because each of these distinct cell types doesn't use the entire blueprint. It doesn't need the instructions for building every part of the house. Instead, each cell type uses only a specific subset of the roughly 20,000 protein-coding genes within that genome.
Okay, so it's selective reading of the plan.
Precisely. And this leads to a really profound truth. You have only one genome, but you possess at least 400 different epigenomes. The epigenome is that crucial layer of instruction, that specific set of directions that ensures a kidney cell focuses only on its kidney functions and doesn't accidentally start trying to behave like a liver cell.
Ah, okay. So the epigenome dictates which parts of the master blueprint are actually open and accessible for reading in that specific cell, and which parts are kept closed off, sort of silent.
So you've got it. That's the core concept.
So if the genome is the fixed blueprint, the hardware, then epigenetics sounds like the dynamic software, the instructions on how to read and use that blueprint, almost like stage directions for the actors.
That's a great analogy. Or like a conductor leading an orchestra, telling different sections when to play and how loudly. At its core, epigenetics refers to changes in what we call chromatin structure.
Chromatin, that's how the DNA is packaged, sure.
Exactly. Chromatin is simply the highly organized way your DNA is packaged inside the nucleus of every cell. It's wrapped around these spool-like proteins called histones, and this packaging isn't static, it's incredibly dynamic.
And that dynamic packaging controls the genes.
Directly. It dictates which genes are turned on, meaning accessible and active, and which are turned off, tightly packed away and silenced. You can visualize this as the epigenetic landscape, a sort of constantly shifting terrain within the nucleus that influences gene activity.
So instead of changing the DNA letters themselves, epigenetics puts marks on the DNA or its packaging.
Precisely. It introduces these molecular marks or modifications to the DNA itself or to the histone proteins it's wrapped around. These marks don't change the underlying genetic code, the sequence of A's, T's, C's, and G's, but they control whether a gene is easy to read and therefore active, or tightly wound up and effectively silenced.
So it's like tiny molecular switches or maybe dimmer switches on our genes,
Ooh.
telling them when to be on, off, or somewhere in between.
That's a perfect way to think about it.
Yeah.
Dimmer switches, and there are two main ways these switches operate. The first major mechanism is called DNA methylation. This involves adding a small chemical tag called a methyl group directly onto one of the DNA bases, specifically the cytosine base, the C. This often happens in regions called CPG islands.
CPG islands.
Yeah, areas rich in cytosine and guanine pairs. When these islands, particularly those near the start of a gene, get heavily methylated, it generally acts like a strong off switch. It makes that gene very difficult for the cell's machinery to read.
Got it. Methylation often means off. What's the second mechanism?
The second major mechanism involves histone modifications. Remember those histone proteins the school's DNA wraps around?
Yeah.
Well, chemical changes can be made directly to these histone proteins. Things like adding or removing acetyl groups or adding or removing methyl groups, slightly different from DNA methylation. These histone modifications can either loosen or tighten the DNA packaging.
Ah, so it's like adjusting how tightly the thread is wound on the spool.
Exactly. If the DNA is tightly wound around the histones, the gene-reading machinery can't get access. If it's loosened up, the machinery can get in and read the gene. So adding an acetyl group, for instance acetylation, often loosens the chromatin and turns genes on. Removing it, decetylation, tightens it and turns them off.
So these modifications, methylation on the DNA, modifications on the histones,
Yep.
they literally control which genes get expressed. And that determines what a cell is and what it does.
Precisely. They determine the cell's phenotype, its characteristics and function, and its developmental state. But the crucial part is they're not fixed.
They can't change in response to signals.
Yes, that's the key. They are remarkably dynamic and responsive. They react to both signals from inside the cell, like changes in metabolism, and signals from outside the cell.
Like from the environment.
Exactly. Environmental cues, pathological conditions like an infection starting, chronic stress, even the very early stages of cancer development. All these things can trigger widespread epigenetic changes. This adaptability allows cells to fine-tune their behavior and function in real time, constantly responding to their surroundings.
Our sources use this really helpful analogy. They talked about writers, readers, and erasers operating on the chromatin. Can you break that down? It helps visualize this complex dance.
It's a great analogy because it captures the active nature of it all. So the writers are specific enzymes. Their job is to add these epigenetic marks.
Putting the marks on.
Right. For example, enzymes called DNA methyltransferases, or DNMTs, are the writers that add methyl groups to DNA. Other writer enzymes like histone acetyltransferases, HATs, or histone methyltransferases, KMTs, add acetyl or methyl groups to histones. They're constantly busy putting these chemical notes onto the chromatin.
Okay, so the writers put the marks down. Then the readers come along. What do?
The readers are another set of proteins. They have specific molecular structures or domains that act like little eyes, designed to recognize and bind to the specific epigenetic marks laid down by the writers.
So they interpret the code.
Essentially, yes. For instance, some reader proteins have what's called a bromodomain, which recognizes acetylated histones. Binding there signals that this region of DNA should be open and active. Other readers might have a chromodomain or recognize methylated DNA via methyl CpG binding proteins like MECP2, signaling that the gene should be kept quiet.
So the readers interpret the marks and then tell the cell what to do with that gene.
They recruit other cellular machinery. Based on the mark they read, they might call in the proteins needed to transcribe the gene into RNA Or they might recruit proteins that further compact the chromatin to ensure the gene stays silent. It's a sophisticated information relay system.
And then there are the erasers. This suggests the marks aren't permanent.
Exactly, and that's crucial for the dynamism. The erasers are enzymes that remove these epigenetic marks. So TE enzymes, for instance, can remove methylation from DNA. Histone deacetylases, or HDACs, remove cetyl groups from histones, while lysine demethylases, or KDMs, remove methyl groups from histones.
So it's a constant back and forth, writing, reading, erasing.
It's a continuous, intricate interplay. This dynamic balance between writers adding marks, readers interpreting them, and erasers removing them is absolutely essential for the precise control of gene expression. It's what allows cells to adapt, to respond to changes, and to maintain their specific functions throughout your entire life. It's a beautifully complex choreography.
Okay, so we have this incredibly dynamic system within every cell, constantly writing and rewriting its instructions. Let's zoom out again. How does this epigenome, this dynamic layer, vary between different people compared to the variations in our actual DNA, our genome? The difference seems quite stark based on our sources.
It is incredibly stark, and this is where the implications for personalized health really start to become clear. If you take two unrelated people, their genomes are remarkably similar. On average, they differ by maybe 4 to 5 million single nucleotide polymorphisms, or SNPs.
Which sounds like a lot, but isn't it a tiny fraction of the whole genome?
It is. It's about 0.15% of the total genomic sequence. And crucially, these genetic differences, your SNPs, are largely static. They're fixed from conception and don't really change throughout your life.
But the epigenome, that's a totally different ballgame.
Completely different. The potential number of variations in an individual's epigenome across all their cells is exponentially larger than the genetic differences between any two people. It's almost unimaginably vast.
Wow. And why is it so much more variable?
Because, as we've been saying, epigenomes are incredibly dynamic and plastic. They vary significantly not just between different people, but also within the same person over time. Your epigenome today is different from what it was last year, or even yesterday, in subtle ways.
And these variations are driven by...
They're a direct response to cellular perturbations, essentially, the cell's unique life experiences. These are triggered by countless factors: your diet, the microbes living in your gut, periods of stress, exposure to environmental chemicals, exercise habits, infections, you name it. Your epigenome is actively recording and reflecting your unique journey through life.
So what does this huge difference in variability actually mean for us? If epigenomes are so much more plastic and variable than genomes, what does that tell us about why people are different, about health, about disease risk?
It suggests a really fundamental shift in understanding. It strongly implies that many of the phenotypic differences we see between individuals, why some people develop allergies and others don't, why twins with identical genes can have different health outcomes, why disease susceptibility varies so much, are often based more on differences in the epigenome than the genome.
So genetics loads the gun, but epigenetics pulls the trigger. Is that a fair way to put it?
That's a common analogy, and it captures some of the essence. Your genetics provide the underlying potential or susceptibility, yes, but the epigenome acts as this constantly evolving interface between your genes and the environment. It's continually integrating signals and fine-tuning gene expression in response. This dynamic layer is arguably a much more powerful determinant of our unique biological responses, our resilience, and our long-term health trajectory than our static genetic code alone might suggest.
That's a really powerful idea right there. Our experiences aren't just shaping our minds, they're literally physically shaping our biology at the molecular level, maybe even more than our fixed DNA.
In many ways, yes. It means our daily choices, our environment, they have a much more direct, tangible impact than we perhaps fully appreciated before.
Which leads us perfectly into our next big topic, epigenetic memory. This isn't just about remembering where you left your keys, is it? Our sources talk about the epigenome as an actual cellular memory system.
It absolutely is, and it's a concept that goes far beyond neurological memory, though it includes that too. The epigenome acts as a sophisticated memory system at the cellular level. It preserves the results of past cellular perturbations. Think of them as significant cellular experiences or adaptations to environmental factors. It preserves these as stable, long-lasting changes in those epigenetic marks.
So changes in DNA methylation, histone modifications.
Exactly, and even changes in the 3D folding of chromatin within the nucleus. These aren't just fleeting, temporary responses. They become persistent molecular records of what that cell or that tissue has encountered and how it adapted. The cell literally remembers its past.
That's incredible. Can you give us some concrete examples? Where do we see this cellular epigenetic memory in action?
Well, let's start with the brain, where we typically think of memory. While we usually associate memory with neural connections, the synapse changes, the neuronal epigenome itself provides the molecular foundation for long-term memory consolidation.
Really? How does that work?
When you form an enduring memory, the actual DNA sequence in your neurons doesn't change. What changes are the epigenetic marks on the DNA and histones within those specific neurons involved in storing that memory. For instance, certain types of DNA methylation marks within the body of genes in neurons are now known to be crucial for the long-term silencing of certain gene expression pathways, which helps stabilize and consolidate memories over time. Your memories are, in a very real sense, written onto the epigenome of your brain cells.
A physical inscription of experience. Wow.
Exactly. But it's not just the brain. Think about your immune system. It has a remarkable memory.
Like when you get a vaccine or have an infection.
Precisely. Your adaptive immune system, T cells, B cells, remember specific pathogens, but even your innate immune system, the first line of defense, exhibits a form of memory called trained immunity.
Trained immunity.
Yes. After an initial encounter with a microbe, innate immune cells like macrophages can undergo epigenetic reprogramming. This leaves them in a heightened state of alert, allowing them to respond more quickly and strongly not just to the same microbe, but even to unrelated pathogens later on. It's a non-specific memory mediated by epigenetic changes.
Fascinating. What about other tissues?
Metabolic organs show this too. Tissues like your skeletal muscle, your fat tissue, your liver, they seem to remember long-term lifestyle choices, especially related to diet and physical activity.
Oh, so.
For example, a prolonged period of eating a specific type of diet can leave lasting epigenetic marks in liver or fat cells. These marks can alter how those tissues handle nutrients, store energy, or respond to insulin, potentially for years, even if the diet changes later. It influences your metabolic flexibility and long-term risk profile.
And what about just basic cell identity? We said earlier epigenetics stops a kidney cell from becoming a liver cell. Is that a form of memory too?
Absolutely. It's perhaps the most fundamental type of epigenetic memory. It refers to the incredible stability of cell identities. Once a cell differentiates into a specific type, muscle, skin, bone, it generally stays that way for its entire life and passes that identity onto its daughter cells when it divides.
How is that memory maintained?
Through very stable, preserved patterns of gene expression locked in by robust gene regulatory networks. Key players here are specialized chromatin-modifying complexes, particularly groups called polycomb and trithorax complexes.
Polycomb and trithorax.
Yes, they work antagonistically. Polycomb complexes generally maintain silenced gene states, while trithorax complexes maintain active gene states, often through specific histone methylation marks. These marks create a mitotically heritable memory, meaning they are copied and passed down during cell division across hundreds of locations in the genome. This ensures that when a specialized cell divides, its offspring inherit the correct epigenetic instructions to maintain their proper identity and function. It's essential for tissue stability.
Okay, so that sounds very stable, almost permanent for a cell's identity. But we also talk about the epigenome being really dynamic, changing day to day. How do those two things fit together? Are there different timescales for epigenetic changes?
Yes, definitely. There's a clear distinction. Short-term, more transient day-to-day responses are often mediated by changes that are easily reversible, especially changes in histone acetylation levels.
Acetylation.
Yeah,
Adding those acetyl groups that loosen chromatin.
exactly. Acetylation and deacetylation can happen very rapidly. Enzymes add the marks, others remove them quickly. This allows cells to swiftly turn genes on or off in response to immediate signals like a sudden change in nutrient levels or a brief stress signal. It's like using a whiteboard for temporary notes, easy to write, easy to erase.
And the long-term memory, like cell identity.
Those long-term decisions, like cellular differentiation or consolidating that trained immunity, are typically stored using more stable epigenetic marks. This often involves specific patterns of histone methylation, which are generally harder to remove than acetylation, and also DNA methylation. These marks provide a more robust, long-lasting memory. The polycomb and trithorax systems, for instance, rely heavily on stable histone methylation to maintain those silent or active gene states over many, many cell divisions. It's more like carving something in stone or writing in permanent ink.
Okay, so the epigenome is constantly changing, but it also builds up this memory over time. This idea of epigenetic drift, that phrase, really captures the lifelong accumulation of experience.
It does. Epigenetic drift describes this gradual, lifelong recording of information of experience by our somatic cells and tissues. It manifests as slow but steady changes in epigenomic patterns, especially DNA methylation maps, as we age and encounter different things. And these changes get passed on to daughter cells when cells divide.
But then there's this next level, this really mind-blowing concept, transgenerational epigenetic inheritance. Are you seriously telling me that what my grandmother ate or how stressed my grandfather was could potentially leave an epigenetic mark on me?
That is precisely what the evidence is increasingly suggesting, and it's one of the most exciting and, frankly, debated areas in epigenetics. While the epigenetic drift in our body's somatic cells affects us directly, the really profound question is what happens in the germ cells, the sperm and egg?
The cells that create the next generation.
Exactly. The idea is that some of the epigenetic marks acquired in response to the environment or lifestyle of the parents or even grandparents might somehow escape the usual reprogramming processes that occur during germ cell development and early embryonic life and be transmitted to the offspring.
So it's not a change to the DNA sequence itself getting passed down, but a change in the instructions for reading that DNA.
That's the hypothesis. It suggests that lifestyle factors of ancestors, perhaps severe famine, significant stress, exposure to certain toxins, could subtly influence the health predispositions of subsequent generations without any change in the underlying genes.
Are there solid examples of this in humans?
The evidence is strongest for effects mediated through the mother during pregnancy, what we call intraschroderine exposures. That's technically intergenerational, affecting the direct offspring. For example, as we mentioned, if a mother experiences significant under- or overnutrition during pregnancy, this can leave lasting epigenetic marks on the fetus that increase the risk for obesity, type 2 diabetes, and metabolic syndrome decades later in the child's life.
That makes sense, the direct exposure in the womb.
Yes. Proving true transgenerational inheritance effects passing through the male line, or effects showing up in grandchildren who weren't directly exposed in the womb, is much harder in humans due to the long generation times and confounding factors. But compelling evidence exists in animal models, and human epidemiological studies provide suggestive links, for instance, between grandparental food supply during their own early development and the cardiovascular health of their grandchildren.
Wow. It's a truly profound idea that the echoes of our ancestors' lives might resonate in our biology today.
It dramatically expands our view of inheritance beyond just genes, incorporating this dynamic layer of information shaped by experience.
And this conversation about accumulated experience, about marks left over time, leads us directly to the ultimate biological clock, aging. Our next section delves right into aging and the epigenome. It feels like a direct continuation.
It absolutely is. Aging is, you could argue, the cumulative result of a lifetime of these cellular experiences and the epigenetic changes they entail. And critically, aging isn't just getting older, it's the single biggest risk factor for nearly all major non-communicable chronic diseases.
Right, you're talking cancer, heart disease, diabetes, dementia.
Exactly. Cardiovascular diseases, type 2 diabetes, most cancers, neurodegenerative conditions like Alzheimer's and Parkinson's, osteoporosis, frailty, arthritis, sarcopenia, muscle loss, macular degeneration, kidney failure. The list goes on. Aging represents a progressive decline in physiological function and resilience across the board, making us vulnerable.
And we've gotten much better at increasing lifespan. People are living longer on average.
True, human lifespan has increased significantly, especially over the last century.
But our health span, the number of years we live in good health, free from chronic disease, that hasn't really kept pace, has it?
Unfortunately, no. There's often a significant gap. People are living longer, but they're spending more of those later years suffering from one or often multiple chronic illnesses. There's a growing burden of morbidity.
Which creates a huge need for prevention, right? Not just treating diseases when they arrive, but stopping them earlier.
Precisely. It highlights an urgent need for effective preventive strategies and promoting healthy lifestyle choices, ideally starting from a young age. The goal should be to compress that period of illness and decline towards the very end of life, maximizing the years lived in good health. And understanding the epigenetics of aging is absolutely key to achieving that.
Okay, so how does epigenetics help us understand aging? I'm really intrigued by this epigenetic clock concept. It sounds like we can actually measure biological age using these epigenetic marks.
That's exactly what it is, and it's been a revolutionary development. The epigenetic clock uses the pattern of DNA methylation at specific sites across the genome to provide a remarkably accurate estimate of biological age.
More accurate than just counting birthdays, chronological age.
Often, yes, it reflects the cumulative impact of lifestyle, environment, and disease on your cells and tissues. Someone might be 50 chronologically, but their epigenetic clock might read 60 if they've had a lot of biological stress, or maybe 45 if they've maintained excellent health. It gives us a molecular readout of the aging process itself.
So it's a biomarker for how well or how poorly we're aging at a cellular level.
Exactly. It's proving invaluable for research into aging, for identifying factors that accelerate or decelerate it, and potentially down the line for assessing the effectiveness of anti-aging interventions.
So what are the specific epigenetic changes that happen as we age? What defines the epigenetic hallmarks of aging and senescence, that state where cells stop dividing?
There are several characteristic changes. We see a gradual loss of histones overall, which can lead to a less organized chromatin structure. There's also a growing imbalance between activating histone marks, like acetylation, and repressive histone marks, like certain methylations, disrupting the fine-tuning of gene expression.
And changes in DNA methylation, too.
Yes, quite distinct patterns. We typically observe what's called global hypomethylation, an overall decrease in methylation across the genome, particularly in repetitive regions, which can contribute to genomic instability. But at the same time, we see focal hypermethylation-specific CpG islands, often near the promoters of important genes like tumor suppressors or developmental regulators, gain methylation and become silenced inappropriately.
So some areas lose methylation, others gain it inappropriately. It sounds like the whole system becomes less precise.
That's a good way to put it. Precision is lost. We also see changes in gene expression patterns reflecting this, losses and gains in heterochromatin, the tightly packed, usually silent chromatin, sometimes a breakdown of the nuclear lamina, the structural support inside the nucleus, and general chromatin remodeling leading to a less organized state.
And these changes aren't just happening randomly. They're influenced by our lives.
Heavily influenced. Environmental stimuli, stress, and especially nutrient availability play a huge role. This is because, as we touched on earlier, the enzymes that write and erase these epigenetic marks often depend on key intracellular metabolites as cofactors.
Like acetyl-CoA, NAD plus MET.
Exactly. So your metabolic state, driven by your diet and lifestyle, directly feeds into the chemical reactions that shape your epigenome and consequently influences your rate of epigenetic aging. It's a direct molecular link.
This connection between aging, accumulated changes, and disease risk seems especially strong for cancer.
Absolutely. Aging is fundamentally linked to an accumulation of genetic mutations over decades. Our DNA repair mechanisms become less efficient, cells divide many times, increasing the chance of errors. This accumulation of mutations, combined with those detrimental epigenetic changes we just discussed, like silencing tumor suppressors, directly leads to the exponential increase in cancer incidence we see with age.
And other things decline with age, too, like stem cell function.
Yes, the addition rates and regenerative capacity of our stem cells tend to decline, impairing tissue repair. Our overall physiological fitness diminishes. Genome instability, the increased tendency for DNA damage and mutations, is a critical hallmark shared by both aging and cancer. It's a fundamental breakdown in cellular maintenance.
It seems like there's a complex relationship here. Cancer cells find ways to become immortal, bypassing normal aging limits, right?
In a very specific and dangerous way, yes. A key step is overcoming telomere shortening. Telomeres are protective caps on chromosome ends that shorten with each cell division, acting as a kind of cellular clock.
When they get too short, the cell usually stops dividing or dies.
Correct. It triggers senescence or apoptosis. It's a natural barrier against uncontrolled proliferation. But many cancer cells find a way around this, often by reactivating an enzyme called telomerase, encoded by the TERT gene.
And telomerase rebuilds the telomeres.
It does. It allows the cancer cells to maintain telomere length and achieve limitless replicative potential immortality in a cellular sense. But here's the twist. While this helps them bypass one aspect of aging, replicative senescence, the process of reactivating telomerase, or the resulting immortalization, often comes at the cost of increased genome instability. It can lead to more mutations, chromosomal abnormalities, making the cancer more aggressive.
So it's a dangerous trade-off for the cancer cell and for the body.
It really illustrates this delicate balance. The body has mechanisms like senescence to protect against cancer, but these same mechanisms contribute to physiological aging over time. Disrupting that balance, as cancer does, has devastating consequences. Finding ways to maintain cancer protection without accelerating aging is a major goal.
This leads us perfectly into something very practical: nutrition. Our next section is nutritional epigenetics, food as information for your genes. This implies our diet isn't just about calories and basic building blocks, but it's actually sending complex signals to our genes via the epigenome.
That's precisely the idea, and it's a rapidly growing and incredibly exciting field. Nutritional epigenetics explores how specific compounds derived from our diet directly interact with and modify our epigenome. It's moving far beyond just thinking about macros and calories.
So specific molecules in food can act like epigenetic drugs almost.
In a sense, yes, they can modulate gene expression without changing the DNA sequence. Think about the vast array of secondary metabolites found in plants, things we eat every day in fruits, vegetables, teas, spices, herbs.
Like what kind of compounds?
Well-known examples include genistein from soy, resveratrol from grapes and red wine, curcumin from turmeric, EGCG and other polyphenols from green tea, compounds in coffee, cocoa. The list is long. These molecules can directly influence the activity of transcription factors, which bind DNA to control gene expression, and also the activity of those chromatin-modifying enzymes, the writers, readers, and erasers.
So they can tweak the epigenetic landscape.
Exactly. They can influence DNA methylation patterns or alter histone modifications. And researchers are using advanced omics technologies, genomics, transcriptomics, epigenomics, to map out precisely how these dietary compounds affect gene expression patterns genome-wide and evaluating their potential for preventing or even helping to treat diseases like cancer.
And you mentioned earlier this direct link between metabolism and epigenetics, through those intermediary metabolites acting as cofactors for the enzymes.
Yes, that's a fundamentally important connection. Many of those key chromatin-modifying enzymes absolutely require common metabolites as co-substrates or cofactors to function.
Can you give some examples again?
Sure. Acetyl-CoA, which is central to energy metabolism derived from carbs, fats, and proteins, is the essential acetyl group donor for histone acetyltransferases, HATs. So acetyl-CoA levels directly link nutrient availability to acetoacetylation and gene activation. Similarly, alpha-ketoglutarate, another metabolite from the Krebs cycle, is needed by certain demethylases, including tech enzymes that remove DNA methylation. And NAD plus dicing, crucial for cellular redox balance and energy status, is the required cofactor for the sirtuin family of deacetylases. ATP, the main energy currency, is also used by some chromatin remodeling enzymes.
So our metabolic state, reflecting what we've eaten and how much energy we have, is constantly feeding information directly into our epigenetic programming machinery.
It's a direct biochemical link. Your diet literally fuels the enzymes that shape your epigenome in real time.
Which connects directly to how our bodies sense nutrients and how that relates to aging. What are the key nutrient-sensing pathways involved?
Our bodies have evolved these really intricate systems. For sensing nutrient abundance, particularly glucose and amino acids, there are two major pathways. The insulin IGF signaling pathway primarily senses glucose. High glucose leads to insulin release, signaling cells to take up energy and grow.
And the other one senses amino acids.
Yes, the TR pathway target of rapamycin senses amino acid concentrations. When amino acids are plentiful, TR promotes protein synthesis, cell growth, and proliferation. Both of these pathways, when chronically overactivated, say by constant overnutrition, are strongly linked to accelerated aging and metabolic diseases like type 2 diabetes.
Okay, so those sense times of plenty? What about sensing scarcity, low energy states?
For that, we have key sensors that generally promote conservation and repair, counteracting the growth signals. The sirtuins, or SIRTs, are a family of NAD plus obus-pendant deacetylases. They become active when NAD plus levels are high relative to NADH, which usually signals a lower energy state or increased energy expenditure.
And sirtuins are linked to longevity.
Very much so. They influence DNA repair, stress resistance, inflammation, and metabolism in ways that promote cellular health and longevity. The other major low-energy sensor is AMPK, AMP-activated protein kinase. It gets activated when cellular energy levels drop and AMP levels rise relative to ATT.
And AMPK also promotes healthier aging.
Yes, AMPK often acts to inhibit energy-consuming processes, like those driven by 2, and activate energy-producing pathways like fat breakdown. It promotes catabolism and cellular maintenance. Interestingly, AMPK can directly inhibit the 2 pathway, showing how these systems are interconnected to balance growth versus maintenance.
So it's this constant balancing act. Insulin, IGF, and 2 pushing for growth and storage when nutrients are abundant versus sirtuins and AMPK promoting breakdown, repair, and conservation when nutrients are scarce.
That's the core dynamic. It reflects our evolutionary history, adapting to periods of feast and famine, anabolism versus catabolism. This balance is crucial for health, and tilting it too far towards constant anabolism, as often happens in modern lifestyles, seems to be a major driver of age-related disease.
This directly explains the interest in calorie restriction, doesn't it? Reducing food intake to tip that balance back towards maintenance and repair.
Exactly. The scientific evidence, especially from model organisms like yeast, worms, flies, and rodents, is incredibly strong. Consistently reducing calorie intake without causing malnutrition robustly extends lifespan and health span in these diverse species.
And the pathways involved are conserved in humans.
Yes, the core nutrient-sensing pathways, insulin IGF, TOR, AMPK, sirtuins, and the regulatory principles are remarkably conserved between these model organisms and humans. This provides a strong biological rationale for why calorie restriction or interventions that mimic its effects might be beneficial for human aging.
So the potential for preventing age-related diseases like diabetes, heart disease, maybe even cancer, and promoting healthier aging in humans seems huge here.
The potential is immense. It's one of the most promising avenues for extending health span. Understanding how to safely and effectively modulate these pathways through diet or other means is a major focus of aging research.
What about popular approaches like intermittent fasting or specific diets like the Mediterranean diet? How do they fit into this epigenetic picture?
They fit very well. Think about our evolutionary past as hunter-gatherers. Our feeding patterns likely involved natural cycles of eating when food was available and fasting when it wasn't. Our bodies are genetically adapted to this switching between nutrient intake, usage, and storage.
So intermittent fasting taps into those ancient programs.
It likely does, activating those pathways like AMPK and sirtuins during the fasting periods. However, the long-term efficacy of many fasting strategies probably depends heavily on the quality of the diet consumed during the eating periods.
Meaning fasting alone might not be enough if you're eating poorly otherwise.
Probably not as effective, no. Combining fasting approaches with diets already known for health benefits, like the Mediterranean diet or perhaps the Nordic diet, is likely the most powerful strategy. These diets provide not just a healthy eating pattern, but also a rich source of those beneficial phytonutrients and healthy fats that positively influence the epigenome.
So it's the combination, the timing, and the content of the food.
Exactly.
Yeah.
And we're now getting sophisticated enough to measure the specific impact of different foods or dietary patterns. Individual dietary components, or even whole diets, induce characteristic patterns, dietary signatures in chromatin accessibility, gene expression, protein levels, and metabolite profiles.
And we can measure these signatures.
Yes, using omics technologies in cell cultures, animal models, and increasingly in human studies. This allows us to really dissect how food acts as complex information for our genes, paving the way for truly personalized nutrition advice based on someone's unique epigenetic responses.
Food is information. That's a powerful reframe. And it's not just food. The other major lifestyle factor is physical activity. How does exercise activate our epigenome for healthier aging?
We all know exercise is good for us, right? The link between physical fitness and longevity is incredibly strong. Regular activity demonstrably reduces illness and mortality, but now we're understanding why at a molecular level and epigenetics is central.
So hitting the gym or going for a run is literally changing gene expression through epigenetic marks.
Precisely. Physical activity directly influences the epigenome in ways that actively promote healthy aging. We see tangible changes in the chromatin of skeletal muscles, for example.
Like what kind of changes?
Studies show that exercise can increase levels of specific activating histone marks like H3K36 acetylation in muscle cells. This suggests exercise is turning on genes important for muscle adaptation, energy metabolism, and repair. We also see changes in the location of certain histone deacetylases, HDACs, like HDC4 and HDC5. Moving these eraser enzymes around within the cell can fine-tune gene expression in response to the exercise stimulus.
And these benefits aren't just confined to the muscles themselves.
Not at all. The epigenetic reprogramming triggered by exercise seems to have systemic benefits. It contributes to preventing cognitive decline, improving metabolic health throughout the body, reducing chronic inflammation, all factors that contribute to a longer health span. It's another clear example of how lifestyle choices actively sculpt our epigenetic landscape for better or worse.
It's truly amazing how interconnected this all is. Diet, exercise, aging, all riding on this epigenetic layer. Let's turn now to the darker side, the role of epigenetics in specific diseases, beyond just the general aging process. Cancer is the obvious starting point.
Yes, cancer is arguably the disease where the role of epigenetics is most profoundly understood alongside genetics. It's not just a disease of mutations, it's deeply rooted in epigenetic dysregulation.
How so? We mentioned silencing tumor suppressors.
That's a major mechanism, but it's broader than that. For example, those repetitive sequences in our genome, which make up a large fraction, are normally kept silent by heavy DNA methylation to prevent them from jumping around and causing instability. In many cancers, these regions become hypomethylated, losing methylation, which contributes significantly to the genomic chaos seen in tumors.
So instability increases.
Yes. And then, conversely, you have the hypermethylation, the adding of methyl groups, specifically at the CpG islands located near the promoters of critical tumor suppressor genes. Genes like P16, BRCA1, MLH1 genes that normally put the brakes on cell growth or repair DNA damage get inappropriately silenced by this epigenetic mechanism. This is a classic example of detrimental epigenetic drift contributing directly to cancer.
And mutations can happen in the epigenetic machinery itself.
Frequently, mutations in the genes coding for the writers, readers, and erasers enzymes like DNMTs, TETs, HATs, HDACs, EZH2, a histone methyltransferase, are commonly found across many different cancer types. These mutations directly disrupt the normal epigenetic balance, further driving the cancer process.
But here's the hopeful part again. Because these epigenetic changes are potentially reversible, unlike DNA mutations, this opens up huge therapeutic possibilities, right?
Absolutely. This is the real promise of clinical epigenetics. The fact that most epigenetic modifications can be reversed is a game changer. It's led to the development of epidrugs.
Epidrugs.
Yeah?
Drugs targeting the epigenetic machinery.
Exactly. These are typically small molecule inhibitors designed to target specific chromatin-modifying enzymes. We have drugs that inhibit DNMTs, DNA methyltransferases, trying to reverse that aberrant hypermethylation of tumor suppressors. We have HDAC inhibitors, aiming to restore histone acetylation and reawaken silenced protective genes, inhibitors of histone methyltransferases like EZH2, or dimethylases like KDMs. It's a rapidly expanding field.
And these are actually being used in patients?
Yes, primarily in oncology right now. Several epidrugs like DNMT inhibitors and HDAC inhibitors are approved for certain types of leukemia, lymphoma, and myelodysplastic syndromes. EZH2 inhibitors are used for specific lymphomas and sarcomas. But the field is moving fast, and clinical epigenetics is definitely expanding beyond cancer into other areas.
Like what other diseases have strong epigenetic links where these approaches might work?
Neurological and neurodegenerative diseases are a major area. Many conditions, from Alzheimer's to Huntington's, show disruptions in the balance of histone acetylation, often involving HATs and HDAC. We mentioned Rubinstein-Taybi syndrome, caused by mutations in a HI gene, CRMABP. The resulting low histone acetylation might, in theory, be treatable with HDAC inhibitors to try and restore some balance.
And metabolic diseases. We keep coming back to those.
Yes, type 2 diabetes and metabolic syndrome clearly have significant epigenetic components stemming from both early life programming, those intrauterine exposures, and ongoing lifestyle factors influencing the epigenome throughout life. Targeting epigenetic pathways could offer new ways to improve metabolic health.
What about the immune system? Aging of the immune system? Inflammation?
Immunosenescence and inflammaging definitely involve epigenetic changes. Factors like chronic viral infections, like CMV, seem to drive epigenetic reprogramming in immune cells over time, contributing to their decline in function and the low-grade inflammation seen in aging. Even our individual immunophenotype, the unique makeup of our immune system, is shaped by epigenetic memory of past encounters.
Which brings us to the microbiome. That community of microbes living in and on us seems to influence everything, including our epigenome and immune system.
Profoundly. The microbiome is a major environmental factor shaping our epigenome, especially the immune system. The trillions of bacteria, viruses, and fungi in our gut, on our skin, etc., are in constant dialogue with our cells.
How does this dialogue work?
The microbes produce metabolites that can directly influence host cell epigenetics. They also interact directly with immune cells lining the gut, for instance. This interaction is crucial for educating the immune system, especially early in life.
Early life exposure is key.
It seems to be critical. How a baby is born, vaginally versus C-section, and fed, breast milk versus formula, dramatically impacts the initial colonization of the gut microbiome. This early microbial community plays a vital role in training the developing immune system, helping it distinguish friend from foe and setting the stage for lifelong immune function.
And disruptions to that early microbiome, maybe through antibiotics, can have lasting effects.
That's the concern, and there's growing evidence linking early life dysbiosis and imbalanced microbiome, often due to antibiotic use, to an increased risk of developing autoimmune diseases and allergies later in life. This strongly supports the hygiene hypothesis, the idea that reduced exposure to diverse microbes early on might impair immune development.
It's not just bacteria, right?
Yeah.
Our personal the viruses we carry also plays a role.
Yes, our resident viruses are also part of this ecosystem, constantly interacting with and shaping our immune responses and contributing to our unique immunophenotype. It's forcing a shift away from thinking one microbe causes one disease towards understanding health and disease as resulting from complex interactions within the entire microbial community and its interface with our own epigenome.
This whole deep dive paints a picture of incredible complexity, but also incredible potential for intervention. It really feels like the future is about understanding these molecular conversations and learning how to guide them, which brings us to our final segment, the future of genomic medicine, personalizing your health journey.
Absolutely. We're standing at the threshold of a new era, driven by the power of next-generation sequencing, NGS, and other omics technologies. We're moving beyond just sequencing the static genome towards dynamically monitoring an individual's entire molecular landscape in real time. And this is where initiatives like the Genomic Medicine Program are so crucial.
And this idea of integrative personal omics profiling, or IPOP, seems central to this future. What exactly is that?
IPOP is the concept of combining an individual's baseline whole genome sequence, their genetic blueprint, with repeated dynamic measurements of their other molecular profiles over time.
So tracking their epigenome, transcriptome.
Exactly. Tracking the epigenome, DNA methylation, histone marks, the transcriptome, which genes are being actively transcribed into RNA, the proteome, which proteins are present and active, and the metabolome, the profile of small molecule metabolites reflecting cellular activity. Integrating all this data gives an unprecedented, personalized, real-time view of an individual's physiological state.
Has this actually been done?
Yes, pioneering studies have shown its potential. For example, IPOP analysis on a single individual over several months detected the very early pre-symptomatic signs of type 2 diabetes onset. Based on these specific molecular signals, personalized diet and exercise changes were implemented, which successfully reversed the trajectory towards diabetes.
Wow, true prevention based on real-time molecular data.
That's the power. Early detection and truly personalized intervention, guided by dynamic molecular data, not just static genetic risk.
So this allows for much more precise stratification of people for clinical trials or treatments.
Precisely. Instead of grouping people based just on symptoms or crude biomarkers, we can stratify them based on their unique genetic background and their dynamic molecular responses. This means clinical trials could potentially be smaller, faster, and yield much clearer results because the intervention is targeted to the individuals most likely to benefit.
And complex diseases like type 2 diabetes could be broken down into subtypes.
Exactly. We'll likely move towards classifying conditions like type 2 diabetes into distinct subphenotypes based on an individual's specific genetic drivers, combined with their unique metabolic and epigenetic responses to interventions, like a particular diet or drug. This allows for much more tailored and effective management.
This really feels like it's blurring the lines between nutrition and medicine.
It absolutely is. Nutrigenomics, studying how nutrients interact with our genome and epigenome, is evolving from basic science into a potential diagnostic and therapeutic tool. Understanding how specific foods or dietary patterns impact an individual's molecular pathways will allow us to prescribe nutrition with far greater precision, both for preventing disease and as part of the therapy, potentially alongside traditional drugs.
So food as medicine becomes less of a slogan and more of a data-driven reality.
That's the trajectory. Using diet strategically, based on personal omics data, to maintain health and optimize therapy, potentially reducing reliance on synthetic drugs or making them work better. It points towards a future of highly individualized, healthy lifestyles prescribed the same rigor as medication.
So to wrap up this incredible journey, we've gone from the basic difference between the static genome and the dynamic epigenome, all the way to seeing how our life experiences, what we eat, how we move, the microbes we host, the environments we encounter, are literally writing our health story onto our genes via these epigenetic marks.
That's right. We've explored how this ongoing writing process influences the ticking of the epigenetic clock, shaping how we age and our susceptibility to a whole host of chronic diseases.
It really drives home that our biological destiny isn't solely predetermined by the DNA sequence we inherit. It's profoundly shaped by this adaptable, responsive epigenetic layer.
Which leads to a provocative thought. What if consciously and intelligently shaping your own epigenome through informed, personalized lifestyle choices guided by this new science becomes the most powerful form of medicine we have? The key to not just a longer life, but a significantly healthier and more vibrant one.
The more we decipher these intricate molecular conversations within us, the more precisely we can intervene to nudge them towards health and away from disease. And that really encapsulates the promise of the genomic medicine program, moving beyond one-size-fits-all to truly personalized health strategies, tailored to your unique genetic blueprint and the dynamic story being written on your epigenome.
It's a future focused on proactive health optimization, not just reactive disease treatment.
Thank you for joining us on this fascinating deep dive.
Join us next time on The Deep Dive as we continue to explore the cutting edge of science and its impact on our lives.